now that we've talked about research and measurements and conducting particular types of studies and what we should do with the results at the end with our statistics and all sorts of things, let's bring it back to thinking critically about research, things that we should be considering when running research as well as when we're reading research. What should we be looking for, especially when we're trying to find good research? So we might want to be thinking about something called validity. And validity addresses how well an experimental procedure is actually testing what it was designed to test. Validity overall is basically saying that this was a good experiment and we set out to test thing X and we actually tested thing X. So if you're trying to look at personality and how it influences your behavior, were you actually studying personality, or maybe you were confounded and you studied mood more than personality? So if you had more of something else influencing your results, you would have lower validity. And we can also talk about specific kinds of validity. The first of these is internal validity. And this is going to be related to the definition we just had of overall validity, but internal validity is specifically looking at the degree to which an experiment is supporting clear causal conclusions. Basically, were you studying what you intended to study, and did you get significant results out of it? So when you have a well-designed and properly conducted experiment, you should have internal validity. And if you have internal validity, you can be confident that your independent variable is actually what was causing the differences that you observed in your dependent variable. You have internal validity, you are fairly certain that you found your causality, the thing we've been looking for all this time. So that is what we're looking for with internal validity. Now, what can threaten our internal validity? Something like a confounding variable. And a confounding variable is basically an alternative explanation, something else that's going on. In my example, when I was talking about you're trying to measure personality and some kind of behavior, a confounding variable could be mood. And if you don't specifically try and control for mood, then it can threaten the internal validity of your study. Another threat would be placebo effects. And this can happen mostly when we see that people's behavior starts changing because of expectations. So for an example of a placebo effect, maybe as a kid, whenever you would fall and hurt yourself, your mom would kiss your boo-boo to make it better. And because she told you that her giving you a kiss would make it better, you believed that and thought that you were feeling better. So that feeling better, because you expect to feel better, is a placebo effect. And that same thing happens in psychological research all the time. It also happens in medical research, all kinds of research. So you have to watch out for the expectations of the participants in the research. We also have expectations on the other side of things. So it's not just the participants that have expectations, but our experimenters have expectations as well. And those can lead to experimenter expectancy effects, where basically the experimenter can unintentionally influence the participants of the experiment. And this kind of links back to what we had talked about with demand characteristics, where our expectations start influencing how people behave because they assume that they should behave a certain way. So there's lots of complicated and interconnected things, and it's all made worse by the fact that we're humans and we like to overthink things. But if we want to reduce these expectancy effects, if we want to keep both the experimenter and the participant from expecting anything, what can we do? We could use what's called a double-blind procedure. And in the double-blind procedure, we're saying that basically the participant and the experimenter are both kept in the dark. If they don't know what's going on, if they don't know if they're in the experimental group or the control group, And if the researcher doesn't know if the participant is in the experimental group or control group, then nobody can have any expectations. If we make sure that the people taking part in the experiment don't know what the experiment is about, that can help reduce expectations. Or we can reduce the human element on the experimenter side and use an automated measurement device so that the researchers themselves don't expect anything and don't have that expectation influence what they're measuring. So lots of different ways that we can counteract this. Another thing that we want to consider, so in addition to our internal validity, we can also talk about external validity. So internal validity was that you were testing what you were trying to test and that you got some kind of significant result that's showing us those clear causal conclusions. External validity is more related to generalization. So external validity is the degree to which the results of a study can be generalized to other populations, settings, or conditions. In plain English, it's how well you can take the results of your study and apply it to other cases. So if you studied undergraduate students at the University of Alberta and got a particular result, would that then also apply to undergraduate students at MacEwan University? How about to university students in another country? How about people who aren't university students? If you have high external validity, then you would be able to generalize to other populations, other countries, other situations. It's more broadly applicable. And in order to have that external validity, we want to make sure that we have a representative population. So when we're sampling our population, we want to make sure we're using random assignment. That's one of the best ways to ensure we have external validity. Another thing we want to consider is the idea of replication. And replication is just the ability to repeat a study and get the same findings another time. So say I do an experiment here at the University of Alberta, and I get the results that X and Y are positively correlated. If somebody else runs the same study, but they run it in Nova Scotia, if they also find that X and Y are positively correlated, we can say that they replicated the findings. And that makes us more sure, more certain, that we're giving more support to whatever theory it was that we were testing. So more replication is a good thing. And this replication is especially important in situations of cross-cultural replication. We don't get into it too much in this class, but a lot of research is done in either North America or Europe. It isn't necessarily replicated in other countries, other cultures. And so a lot of our results, we're not sure if they would generalize to non-North American cultures. And especially we have a lot of stuff that's the research is conducted in undergraduate university students. And we don't necessarily know if that if that would apply to people other than university students. So there's lots of different kinds of replication that we need to look for and that we should push for research to happen on, because we don't know if it would generalize. But cross-cultural replication is a big one. When we're also trying to replicate results and run experiments multiple times, we also can run into something called meta-analysis. And this is a fancy statistical method of basically taking lots of different studies that have fairly similar methods, that we're looking at fairly similar things, and you combine the data that they collected for all of those studies to give you a larger sample size, to give you a larger data set. And it actually helps you maybe have more confidence in the significance of the results, because now instead of looking at 100 students in one experiment, maybe now you're looking at 100 experiments that each had 100 students. So a meta-analysis can allow you to have better statistics and get a better picture of what's actually going on. And now instead of breaking this off into its own video, we're going to talk about ethics because there are only just a couple of slides here. So I've hinted about ethics all throughout this chapter, but when we're talking about ethics or ethical standards, We're talking about things that have been designed to protect the welfare of both humans and non-human animals in psychological research. As a researcher, to follow ethics, you have to protect and promote the welfare of your participants. You have to avoid doing harm to participants. You have to make sure you're not carrying out any studies unless the probable benefit is proportionately greater than the risk, which, again, is a complex way of saying that you have to make sure that if your study has any potential for risk, you have to make sure it's worth it. The outcome, the benefits, the stuff that you're going to learn, and the potential benefits to society should outweigh those risks. We also come into informed consent. So this is the idea that the people who are participating in your research have to know what's going on, and they have to give you permission to be part of that study. So if you want to have a bunch of people participate in a survey, you have to let them know that you're going to ask them a bunch of questions, and they have to tell you that that's okay. So you would use either written or oral consent, and you have to make sure that there's no penalty if they refuse to give you consent. So you can't force someone to participate in your research. They have to be able to say no, and there can't be any consequences to that. So you have to make sure that you're not coercing anyone. And you also have to ensure privacy and confidentiality. So you can't share people's names. You can't talk about people participating in your research. Everything has to be kept private and confidential. Now, if you've been following along, you might be saying, wait a second, we've talked multiple times about the fact that you can't always tell people that they're being observed, and they can't give you informed consent if they don't know that they're being observed. So that's where we come to deception. And deception's a pretty big thing in psychology. So sometimes participants are going to be misled about the nature of the research. So maybe in your informed consent blurb where they're telling you about the research, maybe they're going to tell some white lies or not give you all the details about the kind of research that's being done. And that's troublesome. That's controversial because it's not completely informed consent because they're not well informed. But there are situations where it can be accepted. So it can be permitted if there is no alternative available. If this is the only way that you can get an accurate measure of their behavior, then sometimes your ethics committee will allow it. And if you're allowed to use deception, then there has to be what's called a debriefing session after the experiment. So once you've finished the experiment, you have to sit that participant down and explain that there was deception, what the deception was, and why they needed to be deceived. So then you come clean at the end and tell them about the true nature of research. And we can spend all sorts of time talking about ethics. There's so much. I've done an entire 3-hour lecture just on ethics alone, but we'll hit just the very highest of points here. And a lot of our modern day ethics rules and regulations come from the Belmont report, which was done in '79. And this basically came about because there were a bunch of studies, a lot of stuff related to psychology and some medical studies as well, that were just outright unethical. If you've ever heard of the Tuskegee syphilis study, that was a horrendous study looking at the effects of syphilis mainly on poor, mainly Black communities. And that was one of the launching off points to lead us to discussions about ethics. There's lots of other ones that we can talk about, but again, no time for that. But this Belmont report was basically a bunch of researchers, a bunch of scientists sat down and they made a legitimate framework that would talk about the need for ethics and what conditions needed to be met for researchers to conduct ethical research. So they focused on that you have to respect people's right to make decisions for and about themselves without influence or coercion. That's the idea of informed consent and the ability to say no without being pushed. You have to minimize the risks and maximize the benefits. That's the idea that the potential benefits should outweigh the potential risks. It has to be worth more than the risks that the study has. And then a really important one is that the benefits and the risks have to be equally distributed to all of the participants in the study, specifically without prejudice towards any particular individuals or groups. So you couldn't have all of the people in an experimental group of a risky medical study be a particular group of people that you don't like, because then that group is dealing with all of the risks associated with this experiment, but potentially all of society could benefit from it. That's not just, that's not ethical. So that's actually incorporated into the rules that define ethics for us now. And though we don't go into tons of detail about it, there are also ethics for working with non-human animals. So we have APA's code for working with animal subjects. There's some things that aren't the same as working with humans because a lot of stuff doesn't apply to other animals. So things like informed consent aren't necessarily applicable, but they do have some extra things to consider. And so if you're going to be working with animals, then you have to be trained in those methods, and you have to be experienced with working with that kind of animal. You want to make sure that you're not going to be doing more harm than good. You have to minimize discomfort, make sure you're not causing infection or illness or extra pain. Use anesthesia for surgeries. Make sure that you use pain medication if at all possible. And any kind of procedure that could cause discomfort or pain, again, it can only be used as a last resort. So there has to be a massive potential for benefit to outweigh all of those risks, all of that harm. So mostly we can sum all of this ethics stuff up with ethics is a big deal. And I wish that in this class we could spend a lot more time on it because it is massive. There are lots of rules and they are very strict and very heavily enforced. Again, we don't go into tons of this, but every university, every institution that has researchers is going to have an ethics board. And for any researcher to do any kind of research, they need to have approval of that ethics board. We also have peer review before publishing. So outside of our institution, the places that we can put our research also double and triple check that we have ethical approval. And there's accountability. If you ever go against ethics, you will lose your license. You will lose your ability to publish. You will not be able to do research anymore. It's a big deal, and there's a lot to it. And on that cheerful note, we will leave it there for Chapter 2.